Good morning everyone. Our topic for today is all about characterize the phases of the cell cycle and their control points. For today's lesson, we're going to characterize one by one the phases of the cell cycle and their control points. And at the end of the lesson, we will recognize or we will learn about its function and its importance to our um, cell or in our body. So let's start. Cell cycle is the series of events that occur in a cell to prepare a cell division and to produce the two new daughter cells. So yung cell cycle daw po is ito yung series of events o pagkakasunod-sunod ng mga events that occur in a cell or nangyayari po ito sa isang cell para daw po ihanda siya para sa cell division para makaproduce ng dalawang daughter cell. So, meron pong dalawang phases ito. Ito yung interphase at mitotic phase. So, let's define interphase. Interphase is the longest phase of the cell cycle. It is also as the resting phase. Although the cell is at rest, but it is metabolically active. The metabolic activities of the cell can be observed through cell growth, production of nutrients and enzymes, and replication of DNA. So based on this definition, interphase daw po is the longest phase of the cell cycle o pinakamahabang part ng phase or cell cycle. It is also as the resting phase. Ito yung po yung resting phase. Although the cell is at rest or kahit na po ito yung nasa rest, it is metabolically active. So kahit nasa rest siya, ito pa rin ay metabolically active. So, sabi daw po dito, the metabolic activities of the cell can be observed or pwede daw po mapansin ito through cell growth, production of nutrients and enzymes, and replication of DNA. So, dito daw po napapansin yung mga metabolic activities. And, let's proceed. So, meron pa itong three main stages. Ito yung number 1, G1 phase or GAP1 phase or tinatawag din itong growth phase. And pangalawa, synthesis or the synthesis phase. And pangatlo, the G2 phase or the GAP2 phase or we're also calling it as growth 2 phase. So let's define the growth 1 phase or G1 phase. It occurs about 9 hours. So nangyayari po ito in 9 hours. So the cell increases in size. Photosynthesis also occurs, enable the cell to increase the amount of cytosol. It also prepares for DNA replication. So, based from po dito, ito po yung the cell increases in size. So, dito na po lumalaki yung size ng cell. And photosynthesis also occurs, enable the cell to increase the amount of cytosol. So, meron daw po rito nangyayaring photosynthesis na kung saan pinapataas niya yung amount ng cytosol. There is also a GO phase shown branching off. These represent cells that exit the cycle and stay in resting or non-dividing state, like nerve cells. So based from this picture po, makikita po natin dito yung protein synthesis and organelles are produced and increased volume of the cytoplasm. Then, ayun. Let's proceed. So, pangalawa, synthesis. So, duration about 10 hours. So, mas mahaba to kaysa doon sa G1 phase. So, DNA replication or DNA synthesis occurs here. So, dito na po nangyayari yung DNA replication. Each chromosomes is duplicated. So, the cell now has 2 copies of genetic material. By the end of S phase, every chromosome consists of 2 sister chromatids. So, based from this picture, cell duplicates its DNA. So, kung papansin po natin dito sa definition niya, each chromosome daw po is duplicated. So, nagiging doble. So, the cell now has 2 copies of genetic material. So, syempre, pag nag-duplicate yung isa, yung mga traits and also yung mga contents inside that DNA is also replicated or we call it chromosome. So, syempre, pag duplicate, may same yun na identities or same na laman pag copy kasi na-duplicate niya siya. So, naging dalawa. So, parehas po silang mayroong parehas na genetic material. So, ayun. So, sa G1 phase, it prepares for DNA replication and sa synthesis, dito na lang nangyayari yung DNA replication. And the next one or the last one is the G2 phase or the gap2 phase. So, duration 4 and 1/2 hours. So, the cell continues to grow and produce the proteins needed for mitosis. The final stage where the cell prepares itself for cell division, process, and also ensures that the DNA replication completes. So based from this picture, the organelles are produced and increased volume of cytoplasm. So ayan, dito po nangyari yan sa G2 phase. So sabi daw po dito, the cell continues to grow. So habang naduduplicate, yung mga naduplicate na yan, lumalaki siya. And nagpo-produce ito ng maraming proteins na kinakailangan para sa process ng mitosis. So, ito daw po yung final stage where the cell prepares itself for cell division process. Yan, dito daw po nag-prepare yung cell para po sa cell division. And also that ensures the DNA replication complete. So, ito po yung nag-ensure para po maging complete yung DNA replication na nangyayari doon sa synthesis phase. So, ayun. So, now, let's proceed to the second phase of cell cycle. So, we call it mitotic phase. So, the mitotic phase is a stage of the cell cycle where the nucleus divides and the duplicate chromosomes are separated into two identical sets. It has a duration of 1.5 hours, shortest. This process, also known as mitosis, consists of four main stages: the prophase, metaphase, anaphase, and telophase. So, based from this observation, so dito na po nangyayari yung pagdi-divide ng nucleus and the duplicated chromosomes. Dito na po sila nasa-separate into two identical sets. So, parehas. When we say identical, pareho. And also, ito daw po yung may pinaka-shortest na time kasi po, siya lang ay nagko-consist ng 1.5 hours. So, ayun. So, ibig sabihin, yung interface, kinonsume niya na yung 23 and 23.5 hours na process. And si mitosis phase or mitotic phase, it only consists of 1.5 hours. So, now, let's proceed to prophase. So, short discussion lang po since we're just recalling. So, prophase, chromatin fibers condense and become visible as chromosomes. So, nakikita niyo po sa picture na nagiging siyang, yun, yung mga fibers, nakita niyo yan. Each chromosome consists of two sister chromatids attached at the centromere. So, nakikita niyo po dyan sa picture, yan po yung green yan, yung attached to centromere. Yung dalawa po dyan sa may, dito right side. And the mitotic spindle begins to form. Yung nakita nyo naman yung spindle, unti-unti siyang lumalaki and nagpo-form. And the centrosomes moves to opposite poles of the cycle. So, yan, nag-move po yung dalawang centrosomes at the opposite pole. Mayroon sa north, papunta sa north, and mayroon papunta sa south. Ayan. Next, the metaphase. Chromosomes are aligned at the metaphase plate, an imaginary line in the middle of the cell. Spindle fibers are attached to each sister chromatids. So, yung sister chromatids po, yan yung black, yung red and blue. Yung spindle fibers po, yung nasa taas and nasa baba. Yun po, naka-attach po siya doon sa sister chromatid. Tapos may imaginary line daw po dyan sa gitna, at the middle of the cell. Nakita niyo po, nag-align talaga siya dyan sa metaphase. So, dito po sa prophase, nasa gilid siya. Then, pagdating sa metaphase, sa gitna na po. Yan. Then, the next one is anaphase. The sister chromatids separate and each is now consists a full chromosome. The chromosomes are pulled toward opposite cell poles of the cell by the spindle fibers. Cytokinesis or the physical separation of the cytoplasm begins during this phase and continues into telophase. So, in this phase po, the sister chromatids separate. So, and is now considered a full chromosome. So in this phase po, yung figure po natin, nakikita nyo naman na unti-unti na siyang nagsiseparate. So from metaphase, yan, bilog siya, then anaphase, unti-unti na siyang nagsiseparate. So the chromosomes are pulled toward opposite poles of the cycle by the spindle fibers. So nasa taas, ito yung spindle fibers, and pinupull niya po yung yung mga tinatawag nating sister, tinatawag natin na yun, yung sister chromatids daw po yun. Yan, pinupull niya po yan sa anaphase. And cytokinesis, or the physical separation of the cytoplasm, begins. So dito na po nagsisimula yung cytoplasm. So during this phase and continues into telophase. So ito na po. Next na po, ito yung continuation ng anaphase, which we called the telophase. So the chromosomes arrive at opposite poles and begin decondense. So new nuclear envelopes form around the two sets of chromosomes, creating two new nuclei. So nakikita niyo po yan na the chromosomes arrive at opposite poles and begins to decondense. So nuclear envelopes form around the two sets of chromosomes, creating two new nuclei. So nakita niyo po yan na dito po sa anaphase, oblong. Then pagdating sa telophase, para na siyang dalawang itlog. Pero hindi pa siya totally separated. Parang naka-attach pa rin siya sa isa't isa. Pero po, unti-unti na siyang nagpo-form ng dalawang nuclei dito po sa telophase. So, dun sa anaphase, oblong, and pinupul pa lang po. And dito po sa telophase, mapupul na siya to each other, pero may konti pa siyang hindi pa siya fully developed na pull. Pero may dalawa na siyang bagong nuclei. And the last one, the cytokinesis. Ito na po yung final stage final stage of cell division, where the cell cytoplasm divides to form two separate daughter cells. This process follows mitosis or meiosis and ensures that each new cell receives a complete set of organelles in addition to its nucleus. The mechanism of cytokinesis differs between animal and plant cells. Animal cells pinch inward to form a cleavage furrow, while plant cells build a new cell plate to create a new cell wall. So nakita niyo po yun sa cytokinesis, nag-form na siya ng dalawang daughter cell. So dito po sa telophase, yan, may dalawa na siyang nuclei, pero po hindi pa fully separated. Then pagdating dito sa cytokinesis, fully separated na po siya. So nakita niyo, nag-form na siya ng dalawang cell. And as the time goes by, yan pong mga replicated or dalawang cell na yan, dadami pa siya ng dadami, which is continuous lang po na nangyayari yan. Patuloy silang mag-duplicate ng mga cell na kailangan ng ating body or ng other parts of our ganun. So, nasabi po dito na the mechanism of cytokinesis differs. So, magkaiba daw po sa animal and plant cells. So, animal cells pinch inward to form a cleavage furrow. So, dito daw po, inward pa, ganin siya. To form a cleavage furrow. So, yun. And while plant cells builds a new cell plate to create a new cell wall. So, dito po sa plant cell daw po, nagbibuild siya ng new cell plate to create a new cell wall. So, yan po. And we will going to discuss their checkpoints. So, cell cycle checkpoints are crucial for preventing errors during cell division by ensuring that each phase is completed correctly before the next begins. They function as quality control systems that check for issues like DNA damage or improper chromosome alignment, and either halt the cycle for repairs or trigger programmed cell death or apoptosis if the problem is irreparable. This process is essential for maintaining genomic stability and preventing the development of diseases like cancer. So, mahalaga daw po yung checkpoints kasi daw po, malalaman natin yan dito sa next na discussion. So, in eukaryotic cells, there are three major checkpoints that control the cell cycle process. They are one G1 checkpoint at the G1 and S transition or the synthesis transition. The next one is the G2 checkpoint at the G2 and mitotic transition. And the last one is spindle checkpoint and transition from metaphase and anaphase. So, ayun. So, let's discuss. So, as you can see in the figure po, makikita na po dyan yung mga checkpoints ng checkpoints ng cell division. Ayan po. So, as you can see, may mga naka-designation talaga dyan na para lang, yung restriction or checkpoints sa kada cycle. So, the first one checkpoint is the G1 checkpoint. So, G1 checkpoint checks the following. A, cell size. This does the cell large increase its size or large enough for cell division? B, nutrients. Does the cell have enough reserve energy and nutrients for cell division? C, DNA integrity. So, is any part of the DNA damaged? And D, molecular signals. Does the cell receives growth factors and other signals from neighboring cell? So, as you can see po, may mga nilagay tayong questions kasi po, dito nasasagot yung importance ng mga checkpoints. So, A, cell size. Dito daw po chine-check kung tama po or hindi tama yung cell size. So, yan, does the cell large increase its size or large enough for a cell division? So, this ensures the correct and also the appropriate cell size of the cell. So, pangalawa, nutrients. It also checks the nutrients of that cell. Then C, DNA integrity. Is any part of the DNA damaged? So, chine-check dito yung damage ng DNA. Or kung may malfunctions ba or any problems inside the DNA. And also molecular signals. So, does the cell receives growth factors and other signals from neighboring cells? So, syempre, mahalaga na magkaroon ng signals yung mga cell into their neighboring cell para po walang magkaroon ng malfunction and also problem when the cell division is happening or cell cycle. So, this one, proceed to the next one, the G2 checkpoint. This G2 checkpoint checks the following. So, A, DNA integrity. So, is any part of the DNA damaged? B, DNA replication. Is the DNA replication completed in the S phase? So, as you can see po, sa unang checkpoint, mayroong kapareha dito sa pangalawang checkpoint, which is the G2 checkpoint, which is the DNA integrity. So, sa G2 checkpoint po, ang prior po nito is chine-check niya yung DNA replication. So, ayan. If there is an error, the cell will pause at the G phase and allow for some repairs. If the damage is within the DNA, the cell cycle will pause and let the cell complete the DNA replication or repair it. But if the damaged cell is irreparable, the cell will undergo apoptosis or cell death. It is the self-destruction mechanism of the cell to ensure that the damaged DNA is not passed on the daughter cells, and also important in preventing cancer. There are some cells that never or rarely divided, like matured cardiac muscle and nerve cell, that permanently retains in GO phase. So, sabi daw po dito, if there's error or may pagkakamali, the cell will pause. So, papause daw po muna yung cell cycle at the G phase and follow for some repair. So, na pause and also magre-repair na siya para maayos yung mga error. If the damage is within the DNA or if the damage daw po is mismong nasa DNA, the cell will pause, titigil, and let the cell complete the DNA replication or repair it. But if the damaged cell is irreparable or if the damaged cell hindi na po siya pwedeng ma-repair, the cell will undergo apoptosis or cell death. So, papatayin niya yung sarili niya. Or sinasabi itong self-destruction, ensure that the damaged DNA is not passed on the daughter cells. and also important in preventing cancer. So, yan po. Yun po yung ginagawa niya. Nag-undergo siya ng apoptosis para po hindi mapasa doon sa mga daughter cells that may result in other problems like cancer daw po. There are some cells that never or rarely divided like matured cardiac muscle and nerve cell that permanently retains in GO phase. So, the next one or the last one is the metaphase checkpoint or spindle checkpoint. So, of course, at metaphase stage of apoptosis, it is regulated by anaphase promoting complex or APC. It checks whether all chromosomes are properly attached to the spindle fibers in its alignment at the metaphase plate. If there are mistakes, the cells delays its anaphase process. So, dito na po, chine-check, di ba, na-check na yung mga DNA and other contents doon sa G1 checkpoint, sa G2 checkpoint, and dito po sa may metaphase checkpoint or spindle point, chine-check dito yung alignment ng chromosomes at ng spindle fibers. So, inaano po dito yung proper na alignment. Kung ano ba, kung hindi ba maayos, kung watak-watak ba, kung disarrange, or kung mayroong mga hindi proper na pagkakasunod-sunod or tinatawag natin construction ng isang cell. So, if may magkakaroon daw po ng mistake, the cell delay. So, pwedeng matagalan or pwedeng matagalan po sa anaphase process. Kasi di ba po sa anaphase process, inaayos na doon yung alignment ng spindle fibers at ng chromosomes. So, doon po inaayos sa anaphase process. So, as you can see po in the figure ulit, balikan natin yung figure. Mauuna yung checkpoint or G1 checkpoint, restriction. And also, susunod doon yung G2 checkpoint. And lastly, the mitotic phase or M checkpoint. So, ayan po yung pagkakasunod-sunod. G1 checkpoint, G2 checkpoint, and the last one is the M checkpoint or the metaphase checkpoint. And also, balikan natin yung importance ng mga checkpoints. So, sa G1 checkpoint, chine-check yung cell size, the nutrients, and the DNA integrity, and also the molecular signals. Then, sa G2 checkpoint, chine-check yung DNA integrity and the DNA replication. And lastly, the metaphase checkpoint or spindle checkpoint, chine-check doon yung alignment ng chromosome to its spindle fibers. So, this is other vocabulary in our discussion or in this lesson. So, cancer is the result of the unregulated process of the cell cycle due to the breakdown of the mechanism that controls the entire process. It happens during the synthesis phase, wherein the cell cannot determine the changes in the DNA sequence that code for the specific regulatory molecules. So, the cancer daw po is the result of the unregulated process. So, hindi po regular na proseso ng cell cycle due to the breakdown of mechanism that controls o kinokontrol yung buong proseso. So, nangyari daw po ito during the synthesis phase where the cell cannot determine the changes. So, hindi daw po ma-determine ng cell yung pagbabago sa DNA sequence that code for the specific regulatory molecules. So, syempre, pag hindi na ma-determine ng DNA, kapapasa yun sa mga chromosomes sa iba pang na nagdudulot ng unregulated. So, patuloy lang po na dumadami yung cell. Kaya nagkakaroon ng cancer. Kasi di ba po unregulated, hindi na siya na-determine kung regular ba, kung nasasobrahan or yung katamtaman or kulang. So, yun. So, the next one is The next one is oncogenes. Are the genes or oncogenes? Oncogenes are the genes that cause the cell to become cancerous. So, dito po yung mga genes that cause or nagdudulot ng cell to become cancerous. Ito yung mga sila nagdudulot sa cell para mag-siyang cancerous. Ayun. So, the next one is the proto-oncogenes. Are the genes that code for positive can be an oncogene cycle. When these normal genes are altered by mutation it regulators during cell leading to cancer cells formation. Ito po yung proto-oncogenes, similar siya sa oncogenes. Pero po, these genes code for positive can be an oncogene cycle. So, nagkakaroon daw ng oncogene cycle. When these normal genes are altered by mutation, its regulators during the cell leading to cancer cells formation. So, naaapektuhan daw po ito. Kaya nagkakaroon, or during the cell cycle, yun, nagkakaroon din po ng cancer cells formations. So, sumasabay sila. Nagkakaroon ng cell cycle, and na-altered siya ng mutation. And pag nagkaroon ng mutation, alter mutation yon, the regulators during cell leading, yung mga regulators hindi na po nakakayanan. Kaya, ayun, nagli-lead siya ng cancer cell formation. At yun, oncogenes na yung result nun ng cancer cell formation, which we call oncogenes. Yung mga cell na form that causing to be cell to be cancerous. Then the next one is the tumor suppressor. Genes are healthy normal genes that slow down the cell division, helps to repair DNA mistakes and also cell apoptosis or cell death. It codes for the negative regulator protein. When activated, can prevent the cell from uncontrolled division. But when tumor suppressor gene does not work properly, cell division will be out of control and can also lead to cancer. So, yung mga tumor suppressor daw po are the genes that are healthy, normal genes, mga healthy daw po sila, that slow down cell division, slow yung cell division, that helps to repair the DNA mistakes and also cell apoptosis or cell death. It codes for the negative regulator protein. When activated or pag na-activate na, can prevent or maiwasan yung cell from uncontrolled division. Pero, when tumor suppressor gene does not work properly, so pag hindi daw po ito gumana ng maayos, cell division will be out of control, mawawala na siya ng control. And it can also lead to cancer. Pag nawala na ng control, syempre, hindi na ma-regulate yung yung mga cell doon sa cell division. Syempre, pag hindi na-regulate, kukos siya ng cancer. So, this is the overall review of our lesson for today. So, cell cycle checkpoints are crucial for preventing errors and ensuring a cell divides correctly by monitoring DNA integrity and accurate chromosome segregation before each new phase begins. They halt the cell cycle to allow for the repair of any deleted damage, or if the problem is irreparable, trigger program and cell death or apoptosis to prevent the passing of faulty genetic material to daughter cells. This regulation is vital for preventing mutations and the development of cancer. So, syempre, yung checkpoints, we can also apply it when sa kalsada, syempre checkpoint. So, why we need to conduct checkpoint? So, syempre, checkpoint ensures safety and also para hindi maiwasan yung mga banggaan sa kalsada, para ma-ensure yung safety and also yung mga unregulated na mga pasaway na pasahero na nagkukos ng aksidente. So, di ba, pwede siyang ma-relate dito sa cell cycle and its checkpoints. Kasi minsan, di ba, sa checkpoints, yung mga pasaway na pasahero na walang helmet, tapos yung mga palakad ng motor or yung mga bisikleta is super unregulated. Syempre, nagkukos siya ng damage sa iba. So, same lang dito sa cell cycle. Syempre, pag hindi maayos or yung checkpoint, syempre, pwedeng maapektuhan yung ibang daughter cell or yung ibang contents like DNA. Syempre, makahawaan na yun ng iba pang mutation. So, same lang dito sa checkpoint. So, parang connected siya sa real world and also sa cell cycle. So, thank you very much for listening for our discussion for today. And if you have any questions, you can directly DM me and I will directly respond to that to prevent or to clear clarifications and also to provide more clear and also maayos na explanation sa question na gusto nyong itanong. So, yun lang. Thank you very much and I hope that you gain some lessons. Also, you gain some important knowledge for today. Thank you.